Okay, now let's talk about the next phase of the program. Last month we sent out the questionnaires to our partners. Then we have around how many respondents? 40 respondents. Now, then I will just showcase about the outcome. It will give you some idea for our discussion.
Oh, sorry.
Not working.
You try, no?
Okay. And the first question is, we list out the priority chemicals for the next phase of study. First phase, we done the pharmaceuticals. Then we list out the list on the right, okay? Then turn out the score, how do you calculate the score? 5 is a 7 scale or what?
It's a ranking
Oh,
question.
ranking.
Yeah, maybe people choose first and
This is an average rank, okay?
yeah, average.
The smaller the more urgent, okay? Now, if you will be better to use this one. No battery, I think. Testing better. As you can see here, people like to work on PPCP. I assume that we already finished pharmaceuticals, then we can go for like EDC, phenolic EDC, for example, and other pharmaceuticals, personal care products, like UV filter, and we can think about those. Especially for COVID, maybe UV filter will be important. Okay. Microplastic, the next one. However, microplastic have been well studied, and yeah, then we try not to touch. Okay. Then the third one is PFAS. This is a global concern compound. I think it's worth to go for it, but we need passive samplers to help. Otherwise, we need 30 liters of sample. Cost me a fortune to send 30 liters of sample. So maybe we need passive sampler for this one. Then other legacy pollutants like PAH, PCB, PPD also went forth. Pesticides and the heavy metal, LCM, those compounds from our computer and mobile phone display. Final one is the radionuclides. Those are top on the list, right? Then you can suggest anyone during the discussion. If you're interested to need any of these, then please tell us. And then the second question, do you think a passive sampler would be feasible in your estuaries? Okay, then turn out almost 50% say yes, they have the capability. So right now we have around 100 people, 100 partners whom we send the sampling kits to them already. So if we multiply by 0.5, that means at least we can cover 50 estuaries. Okay, that is good enough, I think, for passive sampler for PFAS. Okay, that is nice. And okay, the next question is, would you be able to collect sediments? Okay, and this is really good result. 80% people say yes. So I think we will move ahead to sample sediment. But we need to work out the optimum samples. The smaller, the more beautiful. But it becomes safer for sending. But we also think about the target chemicals we analyze. But this time with sediments, we can also look at the environmental DNA, antibiotic resistant genes, as well as all kinds of pollutants. Maybe the sample size we can go for 50 grams or 100 grams. And we'll work out how to do it. Then this will be the second part, right?
Yeah.
Should we go now? Or later?
it also okay.
Or maybe I stop here first for the first part, next phase of the GM. Then I open the floor for any suggestions. It's supposed to be a multiple ways of dialogue. Yes, please, gentlemen, and please help to pass the mic.
Yeah, thank you very much for great project. It's already done for the first phase. Actually, related with collaboration, as we sampling for the sample, and then we send the sample to Hong Kong University, City University. There are also another way, because we are not only would like to as sampler. I mean, we only know to sample to sampling the sample, but we also would like to another knowledge how to measure until we get the data. There are any sharing knowledge after that?
That is a nice question. That's why we have to workshop today. I think Luo Chung already presented the method, but I understand your concern. It may be difficult to send everyone to our laboratory to learn. We are thinking to have a workshop for Western Pacific this year for PFAS. We're going to provide some scholarship for people in the region to come to our lab to have a hands-on learning on the skills for PFAS this year. We have yet to think about pharmaceutical. Maybe we should think about this in the near future. But we are thinking to make some video, online training video. We can make step by step so that you know what happened once you get your sample. And I think we can do that with a video to tell people what we have done. Okay, we'll write down. Good suggestion. But we will look for resources to organize a hands-on workshop in the future. Any suggestion, please?
Not a suggestion, but I'm curious about this. It's nagging in my head. It could also affect the sampling scheme. So usually in the papers and also in what we've done so far, it's just surface water samples. And then it's always at the back of my mind that maybe, for example, we don't see some of the antibiotics, or for example, macrolides are much bigger because they are in the sediments. And maybe that's one of the research gaps or knowledge gaps in the literature that we may need to answer, just so technically we've covered everything. So the sediments could answer that, but sometimes the surface water sample or particulates filtered component. Is this also covered in the sampling? Will this also be analyzed?
You are scientists. So in fact, in the river study, we didn't look at the filter. But in this project, we also look at the filter. That means we will also uncover the level inside the suspended solid for the first time. We realized the problem, many lipophilic compound, they will bind to the suspended solid. It's true. And this time we also look at that, right?
Yeah.
So, but we will do it step by step because too many samples to handle. But this is our goal to look at that as well. And you are correct. Many pollutants actually will sink at the seabed. This is the reason why we have the second phase to look at sediment. But for your Filipino study, in fact, you can design to collect sediment and water together. We need to find some way to coordinate the work. Whether your country allows sending out sediment, we have to find out. If you have a coordinated group effort, then we will do the analysis. No problem.
Thank you very much.
Those online also feel free to voice out or type in your question or comments, please. Yes, please.
I would want to comment on the sentiments shipment between the nations. Sometimes it's quite tricky to get permits. Basically because of the legacy of the pollutants that are sensitive to some countries. I think one suggestion to move forward is that instead of sending sediments, like we can do the first stage extractions. Instead of sending the sediments, we just do the extractions in the lab. I think this can be organized and trained. I think it's much cheaper sending a small vial instead of sending that would be my suggestions to move forward. At least that you can skip the parts on sending the sediments. But again, depends on parameters. If you're looking at EDMA, they have different stories, right? Yeah.
This is a good suggestion, but we will need time to do the standardization. And also second problem is the solvent. Of course, you won't tell the airline, they won't understand. But if they find out, it will be big trouble. If you ask the courier service, we have to pay much more money for solvent because they have different container. So I think I will go back to my chemist to think about this. Can you call Phoebe somewhere, ask her to come and then give comment? I have a colleague that's really in charge of the analysis. But this is good for thought. Of course, if the country allows sending sediment will be easier solution, but if not, then we have to go for extraction.
Yeah.
In fact, I won't tell you which country. They send us sample. We done the analysis, and then they told us that we have to throw them away. We cannot publish, even pharmaceuticals.
Yeah.
So it happens. It happens. Yeah. And any comments, questions?
Yeah.
Yes, please.
Yeah, I think the same concern with the Philippines. We really have difficulty sending, substrate and of course, anything with the even powdered, coconut wire. So it involves a lot of clearances and permit from our government. And even though we can send it, later we couldn't publish it. So that's a very thing that we're very much concerned in the Philippines. Even if you send to FedEx, you have to sign on the declaration that this is, not a plant material. And even if it's, environmental DNA, it must not be the DNA, but it should be barcoded. So that's one thing. So I think the suggestion is good that if we have like a common layer that, we can agree that you can develop that could be, applied in many countries.
That's why water is easy. Internationally,
Yeah.
allowed to send water.
Yeah,
That's why we make a first step.
yeah. The filter is good because the filter is,
And also, I think passive sample are also.
I'll just add into that. Sometimes there are ways that we send. I do sediment samples sometimes. I just have to write the certificate that I connected the sediment samples myself at this depth. FedEx usually requires that at this depth, and I assure you it's not a living sample. That's all I'm saying. But sometimes it gets stuck in the country where I send it to.
Hong Kong, no problem.
Okay.
We have free port.
Like in Australia, it gets stuck, but we're able to send it as long as I.
But not mainland China. Hong Kong is still okay. That's why we can do this project.
And then you can apply for, there are several permits that you could apply.
Okay,
Within the
okay,
Philippines.
it's still workable.
Yes.
Okay, good to hear that. We need to have this question when they register for settlement.
Yeah.
study,
Okay.
and they need to go through the legal process.
Yeah.
Any other comments or suggestions? Professor Ong?
May I just
Oh, yes,
follow up?
please, yes, please.
I think the rule in the Philippines is that when you share sample, there should be an agreement between the one that will be accepting the sample. Because the government would like to be assured that if this is a research work, the Filipinos would have a share in the IP, in the intellectual property, if there is one. So one thing that we should be assured of is that there should be an agreement between the receiving party and the one sending the party. I mean, some,
Then we can do that,
Yeah.
elect.
but it's easy for the university, as long as there's an agreement. It's just from the university and, well, I used to be the director of the international linkages of the entire UP system. That's why I had to work, to tell this.
Thank you for your the wisdom sharing, very, very important. So you should talk to her. Professor Ong.
Yes, I agree with every country we have their regulation, especially in sediment. I don't think Malaysia is allowed to sell sediment outside. Because last time I had experience before, because I did my PhD in France. I want to bring back the sediment from France back to Malaysia. I applied officially through the Malaysia, I don't remember the Ministry of what. Okay, what they respond me is they need me to buy ticket from an officer from Malaysia to go to France and then guide me back to Malaysia. So they are not allowed me to, like, for example, like they want to see me to packing the samples, and then they are not allowed me to open the sample until I reach the lab. That's the reason, because they're afraid that they bring some like invasive like bacteria or don't know back to here. And then I have to prepare two sets of samples. One sample will be given to the I don't know what the department to analyze first. Save so I can take all. So since the process is very complicated and take long time, what I did actually I just put in my luggage. So I think I have a 100 sampling site. So I divide my sample to two luggage, because if one start, so I have another samples. So, but the problem when I reach KL, actually they say I bring drug to Malaysia because it's powder form. So I be called, guided by the pistol and everything, to the room, and then they asked me to open my sample. But my wife bring another bag, go over. So I left half sample to the sample. So I'm not sure about this one, because is it possible that we just post the sample with a very minimum, for example, like 2-3 gram sample by the air post?
But still, we have the liability problem. If we publish and then your government find out, then we'll be in a court case, you know. This is something we concern now.
But I think when we publish, I think now the Malaysian concern really is now some researcher from outside, they come to Malaysia for example, and they publish paper without the Malaysian researcher. They're not allowed. Now they are trying to look for those researcher that doing like this, because we have several problems with us country come to Malaysia.
And on the other hand, would it be useful, because I talked to Julia, the director of Ocean Decade, he is here, right? I talked to him yesterday. He said he can write letter of support. Then you can send to your government whether your government support this United Nations project, allow us to receive your sediment sample.
Yes.
This may be a better way.
I think maybe they try.
Because I don't want to be involved in any lawsuit.
Yeah.
I don't have money.
Agree. Because the another thing is someone teach me, he said declare as food. Okay. Because when you declare food, actually it's not really.
They try to voice each sediment, but not me. but it's important to discuss this matter. It is good to know.
Okay.
Okay, any other comments from our online audience as well? Please feel free to speak up. Yes, please, Professor from Thailand.
I'm just curious, you know, when you talk about the eDNA, what did you have in mind from the sediment? Is it microbial communities?
So yes, microbial community is one, but now we can also look at invertebrates and fish as well. So ideally, if we have all the barcode database, we can check the species ID, but we don't need to. We just look at the OTU. We can look at the biodiversity based on the genes. This is an easy way to look at, because we want to have some benchmarking on the ecological status. For example, the high organism, we found 100 OTU, and then in a polluted site, but we found 1,000 OTU in a clean site. Then if we have big sample size in the world, we can look at the correlation. We can benchmark what does it mean healthy or not. This is a simple idea. Recently, we published a paper in ESNT. We found that there is a shift of the microbial community due to eutrophication. There is a tipping point. We can actually see the change based on multiple parameters. Then we can look at in a global context. That would be a very nice study to define the tipping point. Something like that, my colleagues working on microbiomes can help. But we can actually extract lots of information from the eDNA now. For example, we can benchmark some endangered species like seahorse. So whether seahorse can survive in polluted water, we can do that as well. If we have a really big data, and using advanced analysis, we can pick up those signals. For example, we are concerning shark population. We can look at shark population in estuary based on some known biomarker that we can. This is, I mean, gold mine in the future.
Right.
However, like the country won't allow to send out eDNA, then that may be difficult. Yeah. Yes, please. Yeah. Yes, I think we need to standardize the method. I think as DNA sequence now pretty standard. Then once we standardize the sampling, DNA extraction or RNA extraction protocol, we use the same kit, then we'll probably similar result with triplicates, whatever. Then if we follow the same protocol, then we can compare around the world. That may be another solution. Later on, we can share the data, but not the DNA itself. Then it's okay, right? If we look at the data rather than share the DNA materials. That's my point. Yeah, microphone, please.
Guys, my point is, go back to the authorities that they will come back to us when they find out that we send sentiments elsewhere to the analysis, right? They come out is that looking back for the nation, that is most of the concerns. But I think should not stop us to do this project. I think it's a good project. We're talking about globals and standardizing method and get a big picture. What I'm trying to say here is that we start with less sensitive for samples. So eDNA might be the end game still, but we always think the eDNA is important. But for this year, next year, just push for other things like legacy, animal pollutants, pharmaceuticals, that we showcase that looks this is the efforts of global efforts, you know. And by these organizations, and now we have better pictures, can be published, showcased in the UNDK projects. That is actually giving us confidence when we talk to the opportunity, they see this is same was doing the same thing. Now we venture to the next step, EDA. So this is my point, is that if we put everything to the authority that we're going to do EDA, most likely to shut it off. That is my personal view. So yeah, I'm not sure about Thailand and Philippines. That this is, I think, is what is in my mind. That always cautions that building is an example.
Yeah, very good suggestion. Okay. So we will defer the e-learning. Yeah. Yep, step by step.
Maybe it's a top down side. It's a UNDA project, right? So we put the paperwork nicely that for all those partners that can showcase to the authorities, say, this is we are participating in the UNDA project, and each nation will sign that, right? And they agree to support. So in that case, that is back up the projects and get a local support. When come to permits, come to export stuff, they have a proper documents. It's just that they need to be find a way that how to get the document. If you can provide some examples that you have done with certain countries, maybe you can share with all Malaysia and the rest. So see whether it suit the nation needs. If not, then maybe we come back, we need more documentation. Then they will move out. Maybe we can deal with sending the certain examples.
Very good, very good. May I go back to the first question about the list of chemicals we should look at? Yes, the gentleman at the back. Oh, okay. So come back to this list. Do you agree with this? Yes. You can have alternative suggestion. Yes, please. You have comments? You?
Yeah.
Yes, please.
Well, I agree. Yeah, but I agree with what is listed here. So yeah, microplastic, we take them off. Yeah, I mean, the sequence is really nice, the priority. Yeah, PFAS, that's also connected with, plastics, that's component of one of the plastic additives. Yeah, but I have some comments on
Yes,
the
please.
PCB
Yes,
as well.
please.
So yeah, my work in the past has been always linking the presence of pollutant and the microbial community and the microbial genes. And yeah, that's of course the fate, usually the fate of many pollutants are affected, by the microbial function, the microbial community and the microbial dynamics. So since we have this global collection of samples, even from the water samples, I'm thinking it would be a great time as well to, if we can sample, side by side for microbial analysis, then we can analyze this later. Anyway, like I'm thinking, if I'm gonna sample for, this, that I will send you to Hong Kong, I will also get a sample for microbial analysis that I can keep later and I can analyze later. At least at the point of sampling, I can relate, the presence of bacteria to the concentration of the pharmaceuticals in that area. So I think, sampling water or sediment would be also a great time to sample for microbial community and microbial analysis. Even ODUs is good, but now the database has been, substantially enhanced in the past 10 years. From the ODUs, you can predict some genes. And that genes could be a predictor, for example, of the degradation of your PPCPs or PFAS or, shall we say, pesticides. So there are some bioinformatics, amazing or so extraordinary progress in bioinformatics this time that you don't have to analyze the microbial genome. From the OPUs alone, which is much cheaper and much easier, you can predict some genes that could relate to the degradation of some of these pollutants.
Very nice. I tell you what my ideal kind of thinking. You know, we're going to give you the artificial muscle, the perspective too. We're going to have biofilm on it. We initially thought we just give a cotton bud and also the fixing solution for DNA. You just scrap off the surface and then put in the DNA solution, send back the epitope to us. That's it.
Yeah.
So, but however, now we concerned about this secret DNA problem. But to us, it's quite straightforward. We have the sampling device for metal radionuclide, and then you scrap the bacteria. That would be very nice already. We will get lots of information from that. But that would be something like what he did, just put in the other luggage. You know. I bet people won't realize this, but I really worry if some country find out and they're not very happy, then we will be in big trouble. Yeah.
I'm thinking for
Yes, please.
it's quiet, but I honestly, when I analyze, for example, some examples where PEA's analysis in some environment, it really gives some samples for microbial analysis. Anyway, you have that DNA solution that you can keep in your freezer for years, they will be stable. So, I mean, if we need it in the future, at least we have something to support if we need it, if we do the microbial DNA in the future. I mean, for those who can keep this one, and those who have like -20 degrees or -80 degrees that they can keep in their lab. Yeah, just some filters.
Maybe we should follow his suggestion. We have a two-way. Either you do your sequencing in your country or you send to us. We give you choice. As long as you using the same extraction protocol. Yeah. Our senior wants to give comment.
Thank you for, referring to me as a senior. Anyway, no. Anyway, it's not actually, well, anyway, I wasn't here at the start of the presentation, whatever. But I'd like to know whether you will be publishing the results later on, aside from the materials that could be used possibly as pharmaceutical products or whatever. Would you be publishing results and be comparing pesticides, for example, or microplastics from one area to the other? One estuary in another.
Yes, we right now working on pharmaceuticals based on water sample, which is legal, no problem, sending water sample.
Yeah.
Later on, we will try to work on other pollutants.
Yeah.
But we will publish for individual group of pollutants first. But maybe later on, we can do a synthesis paper, put them together.
Yeah, my concern is when the samples are collected in different places and different times, they are not that comparable. And always when there is a meeting and the Philippines is always ranked as the #1 plastic polluter, I always ask them, please give me the details of your methodology. When it was collected, how it was collected, are the different samples comparable? So if the Philippines is ranked as first in plastic pollution, the data should be comparable, but they cannot answer. So I would like to be assured that if you would be comparing, well, I'm a senior already, I might go anytime. But here are the juniors here. I would like to be, if I read the publication, I would like to see a very scientific methodology for comparing data and analyzing data. That's what from the senior.
We have an example already. We've done the 258 meters on pharmaceuticals residues. We publish in PNAS, and we provide all the raw data, including which day they sample and when they sample. And people always be able to get the primary source of information. We will do the same. because this is like a snapshot taking a picture for the whole world. It's hard to organize at the same time, at the same title movement, same weather. It's not possible. We just want to use a standard method to have a global picture. How good or how bad of the pollution situation, and then we can possibly identify some general pattern for management use. That's the idea. Yeah. Yes, please. Lady first.
Yes,
Okay, there's a couple of things. So I sort of understand the point, because I'm sort of thinking along the same way that our countries, if it's ranked really high in something, they may be kind of less receptive of us doing sampling. Because one of the first question I got was, is it going to reflect on the entire country or things like that? So I think your approach about detailing the methodology is something that is of value, and it's sort of like neutral in a way. But in political realms, things get picked out, like in #1 polluter or something. So I think we need to be a little bit careful. But I'm pretty sure that this group has experience in the past, so I'm not too worried. And the second point is that, it seems like there are some methodologies, issues that you like to do workshops eventually. So I would suggest maybe laying out some of the topic, and then maybe sort of pick a location like in Thailand, if people are willing to come to Thailand for certain type of training. So I think if we lay out some of the things of interest in your past experience, like, what kind of training people like, I think I would be willing to sort of co-host.
Yeah, excellent idea. That's it. What is my plan is, once we have the manuscript ready, we will make the PowerPoint ready. Then, for example, in my PowerPoint, I can say we rank from different river whole world. Then you can highlight those Thailand river, where they are in the rank. Then you can say how bad or how good we are. So this is we will make those slides that you can modify for your own use. I think all members will have the same PowerPoint you can use, and you can go for conference. This is your work anyway, and we try to make it very transparent. Everyone can share the ownership. I mean,
Right.
you know,
Sometimes government take things very seriously when
yeah, you're right. That's why a country suddenly pull out from us. But this happened. But I think it depends on how you
Yeah, but if there's
make the
many,
story.
if there are many river, I think that is sort of put in perspective on the rivers, not a country per
Yeah,
se.
we use river instead.
Yeah. And in a country, maybe there's several rivers, so it's not like the entire country is polluted or something. So I think presenting it as, by estuaries, I think it provides perspective of, within a country, there's some heterogeneity or some good example in that country, in some places where there's needs some management. So I think that is a good approach. And
I give you one example. When we report our result for Hong Kong, we are the #1 with most medicines types. We found 43 out of 61, #1 in the world. Our
you
government
already
says
have a champion,
no campaign.
nothing.
They just feel it natural because we're a densely populated community with lots of elderly people. And so, but it triggers some positive action. For
Yes.
example, because the NGO also read the paper, they found out that Hong Kong people used to say dump their unused medicines and expired medicine into toilet and they flush. And then they did a survey, and then they have a campaign to collect all the unused medicine. That really successful. I mean, sometimes it will trigger some positive reaction.
And I have a question, we report the level of everything, but then do you have any link to the toxicity?
Yes, I mentioned in my talk, this is the data gap. For many
Like caffeine.
pollutants, we don't have the PNEC, the predicted low effect concentration.
Okay.
Neither the toxicity data from primary producer like LP to invertebrate to fish. We need at least to have 8 species to build the SSD to find out the PNEC. So this is what we're going to do. We are applying for funding. Right now, Demi did the risk assessment. Many data actually is from simulation using the USCP the program to generate those virtual toxicity data. In fact, it's not too reliable. And so this is one thing we have to deal with. And if you're interested, you're a toxicologist, please join us because we can't deal with hundreds of chemicals just by our lab. If we can have joint venture that can speed up the process. This is why we are here. We need more collaboration with different parties. Yes, please. Yeah.
I agree with what you said, that maybe the focus would be for each country to include in their sampling plan a pristine area, an area that's maybe agricultural, area that's maybe urban, industrial. So that it's a good representation for the country. So I guess it can be planned by each country. And then I guess it's the focus also for the whole sampling plan is to move towards the solution. So then, because, for example, in the Philippines, some of the agricultural areas are owned by multinational companies. And I do agree that looking at it on one end at the start, where we're reporting all these pollutants, it can be very tricky, but on another framework, where we're looking for solutions, it can also push these multinational companies to really find solutions. And there's a lot of countries will look into them to do that solution also. So I think it's just a matter of putting ahead the solution framework.
Can we get donation from those big companies? We need more money for the project. So maybe Chung, do you have any comments, input for this part?
I think our partners all proposed very excellent ideas, and we can collaborate more in future to propose more project, then we can work together and and also solve some questions that we are facing and do every work step by step. I think it's a great idea, and that's my opinion. Thank you.
Okay, so in conclusion for this part, we will follow this recommended list by our partners, and also we will explore the possibility using sediment, and also we will start to prepare the passive samplers, especially artificial mussels is all already mature. We will produce them and then send to any interest party to try it together. Then we can move on to the second part. Sorry, more comment.
Yeah, not fun. So I think this is an amazing group. You really started this. This community to work on this. Do you have like a monthly or once every 2 months like virtual meeting that we can say hi, hello, and update some important neighbors in our respective countries. I think that would be great. If we can have like once in 2 months virtual gathering via Zoom or something.
And maybe 2 months already, high frequent. We can promise to have at least 2 meetings per year,
Okay.
at least 2 meetings. This one we already have, then at least two, right, okay. Now move on, sorry, our, and no, this one I will call it
Okay, that's.
our excellence.
No, never mind, thank you. I was really reacting to your question whether you could have funding from the companies or whatever the computers supposed to be. I remember actually there was a positive response from the people in the Philippines when that plastic ranking was out. And there were some companies, like for example, one cement company gave funding to our institute for collection of plastics in Manila Bay. And so I think once you're out, you can with some data, but you have to be sure that you can defend your data. Then some companies might be able to provide for the solution of whatever problem. And then even the Senate, the legislative in the Philippines, they usually have, like I was able to go to one Senate hearing on plastics, and they called all those companies producing plastics. And they said, if you can reduce the plastic production at at the source, you will be given tax incentives. So the government really responded quickly on that. Although there was, I requested the juniors to proceed with some research to prove or disprove the ranking of the Philippines as #1 leader of the marine environment for plastics. So there's a project, I think, is also involved.
I have to stress that we're not going to make the ranking based on country. We make the ranking based on the river estuary system. Because we only do sampling, we couldn't get the whole picture. We will have this, mark this down, we have to be careful.
Yeah. That was also my thinking, like, according to Madam, so the ranking must not be by country, but by river or something. But in the Philippines, when I was looking at, the ranking of the rivers, like out of the 15 top most polluter of plastics in the world, 11 are from the Philippines. So it's difficult to disprove that. 11 out of 15.
Well, of course, the methodology should be, the rigor of the science should be there.
Okay, let's move on to the next one. In the end game, use your word, we try to provide some recommendation for solving the pollution problem, okay? I just want to give you some initial thinking about how to move forward. We will not be able to have magic bullets to solve the problem. We all know this. But what we are thinking is that we hypothesize that we may find the same pattern as what we found from global river study. Likely, same problem will happen in those lower to middle income countries. Okay, and then we can also try to tease out the best practices, how to minimize the pollution. In some of those countries, will do quite well in terms of management of waste and wastewater. Then we can borrow those examples. For example, instead of open dumping of waste and wastewater, then better to use septic tank. Septic tank, you just need to make a simple concrete tank at your backyard. Then only after the settlement of the solid, the surface water relatively clean will infiltrate to the soil. That's have been used by whole North American, is quite commonly used, already cut down half of the pollutants, for example. Then we can suggest something simple and also compulsory collection of unused expired medicine. That is another doable approach. So what I'm thinking, we should have some tailor-made solution for different country at different level of socio-economic status. And we will make a white paper based on our data sent to UNEP. And also maybe food, the researcher in different country, and you can send to your environmental authority. So this is initial thought about this, how to move forward. Then we don't want to do something like victim blaming. Oh, Philippines, you are the worst, sorry. so then we shouldn't, it's just joking. So I mean, we won't do that because this is the not good because we only have handful of data. We shouldn't jump to those far-reaching conclusion. Then we have to be careful. I think we should look for a big picture when we do the recommendation. Yes.